Welcome to Nikolai's Genetics Lessons, and today's question I got from one of my subscribers. So the topic of the video is going to be why are the bands in my SDS gel frowning. Here is a description of the conditions under which gel electrophoresis was run. I performed SDS-PAGE analysis on rice storage proteins extracted from mature grains using SDS sample buffer. Here's a description of the buffer. I used 14% separation gel with the following electrophoresis conditions: 80 volts for 30 minutes stacking and followed by 120 volts for 120 minutes separating. However, I am observing band frowning specifically near the bottom of the gel, low molecular weight region. The upper portions of the gel appear normal. Could you suggest probable causes for this distortion and recommend solutions? Maybe it's not very clear, but gel wells are here on the top. I'm just going to highlight them. And the bands running from the top to the bottom. And as you see, top bands appear almost normal. But as we move down, now you see at the very end, they start distorting, and this shape we call frowning. By the way, we also have similar distortions, which we call smiling faces, when we have a distortion which would look like this. So this is frowning, this is smiling face. And both of these distortions are caused by the same mistake: too high voltage applied for too long. So advice would be just try using lower voltage. Instead of 120 volts for 120 minutes, you may try using 90 or 100 volts. I also would recommend using cooler system or adding ice gel bags around the buffer with a gel tank while running. Or run the gel in a cool room with pre-chilled buffer and try to use freshly prepared running buffer. You even may try to put tank in the refrigerator while running to cool down the buffer. If the problem is not resolved, then optimize the separating gel concentration and sample volume in each lane. Hope this helps, and this is all for today. Subscribe and see you in the next video. Goodbye.